I've been really grateful to have iSTEAM interns this summer working with me on these very important and high-profile topics and issues. It helps them understand how meaningful the work can be for community, which is our first priority always, but also for the broader public. The work that we're undertaking to use this technology, to communicate those results, to share best practices, really does have real-world impacts. And in my experience, many Indigenous students want to be able to help their community. They want to be able to use those tools that they learn in a university setting to be able to give back. I think that having interns involved in the work we do shows them how that is possible, how they can use that science and use those methods to meet a very fundamental and important need around this very sacred and difficult work. As an Indigenous scholar myself, it is really meaningful to see that next generation of researchers and scholars and scientists emerging in front of my eyes during that summer internship program.
You see those two bees there over there doing their dance?
Yes.
That's how they communicate the food location. And one bee finds a good source of food, they come back to the colony and they dance like that.
I'm participating in the iSTEAM program with Brianna as my student for the third time, and it has been every year a very enriching experience. We have new questions and new studies every year, and Brianna, especially this year, has a wonderful skill set for this experiment that we're doing here in this apiary. It is about egg size and queen behavior, where we had to build small experimental nukes. But we really are trying to push the boundaries of what is known about honeybee reproductive behavior.
This summer I get to work on bee egg size and manipulating different variables to see what changes it, what makes them smaller, what makes them bigger. And I got to help build part of the experiment by coming up with these little nuke boxes here, which had a lot of responsibility with keeping them healthy and making sure that none of the queens die. I've learned lots about honeybees and how to keep them and what different things influence them and what a healthy hive looks like versus maybe a queenless hive. I've also learned lots from my lab mates, just what they're researching and all their excitement and what they've been doing all this time. They've all told me that I should pursue grad school since I seem to be good at this. So that was definitely on my radar before, but now it's definitely a serious consideration. After my internship, I'm heading home to take home some knowledge, and we'll be doing kind of a panel thing at home where I get to display all what I learned and stuff like that, so I'm very excited for that as well. I've actually already reached out to one of our head farmers because he already wanted help with honeybees, so I'm hoping that I can take some knowledge there to help them with pursuing that. So I'm very excited for that as well, and to get to help at home with what I've learned here.
We've got to start our experiment in the lab, so let's go.
I mostly work on laser-induced breakdown spectroscopy for different heterogeneous material, and permafrost is one of them. Aaron mostly work on and helping me with this project to analyze the permafrost to differentiate the low and high organic and inorganic content, carbon content in the permafrost.
The first real thing I did was design a sample holder so we could actually put the permafrost in on the stage and move it around, and also have dry ice in there so that the sample, which is permafrost, can stay cold during our experiments.
He designed that sample holder and designed the experimental parameters like what kind of focusing geometry we need to create the plasma. After that, he perform experiment for different laser energy.
So this was actually the first time I got to do some experiments. It was really cool being able to work with these high-powered lasers and using libs to actually see it in person on a sample. The entire internship opportunity has been amazing to be a part of and has given me a lot of clarification for how Indigenous peoples are present in research and for myself to gain an understanding in what research is. I'm really thankful for iSteam for giving me this opportunity as an Indigenous person to work in the lab and gain some experience that I've never done before. And I also want to give a thank you to them as well for matching me with this project.
In my lab, we're using molecular biology and genomics to investigate tree responses to pests and pathogens. Bo's been working on two different aspects of our larger project where we're looking at how lodgepole pine trees are able to withstand attack by mountain pine beetle.
I've been working with the Jazz gene, which is a plant defense against the mountain pine beetle. I've been looking at the primers that amplify the specific gene. I've learned everything about molecular work and tree care. I've learned a lot of life lessons. I do think this kind of research does really benefit First Nations communities. I would like to see this kind of Western science be applied to traditional knowledge and be a part of that policy making towards traditional lands. And coming to this lab, I came with an open mind and less expectations, but during this internship, it has changed that direction a lot. I do intend to go into grad school. The iSteam Pathways project, it's a very unique opportunity that I've been so pleased to be a part of. You don't really see this kind of opportunity very often.
Our research is conducted on the land. First Nations are the rights holders of that land. They are the stewards of that land. It's important for our personal journeys and our professional journeys to become more connected with the land and the stewards and the right holders of those lands. Bringing iSteam Pathways students into lab is really important to expand our horizons and help us on our journey towards truth and reconciliation. And the iSteam Pathway is just a fabulous program for facilitating First Nations student participation in environmental research. And we're really fortunate that we have this opportunity to connect with First Nations students, to bring First Nations students in the lab through this incredible program.
Has a fragment of a much larger adult mammoth tooth.
These are the grinding surfaces. These are grazers. About a year ago, there was a mammoth that was found just outside of Inuvik in the Mackenzie Delta by some of our northern partners. And it was extremely well preserved in terms of the tusk and some of the postcranial, some of the bones from the skull. And there was a lot of really interesting material, fine-grained sediments that were there that looked to have a lot of plant materials in it. One of the things to do to understand this, because mammoths are very rare in this part of the world, is to look at those plant fossils and the insect fossils from them and see what we could do to reconstruct the environment in the past. And so that was what we developed with Scott and Max as a summer project for the iSteam program. The work that Max has done has shown that it's probably at least 100,000 years old and perhaps quite a bit older than that. So Max has really played an important part in understanding the environment that these mammoths were living in the Mackenzie Delta region, which looks quite different from the environment today and quite different from what we would have expected during the Ice Age.
Max was coming from a horticulture background, so a genuine interest in plants and soils. So this was a cool opportunity to merge our two interests into a research project that would let Max dive into not only the plants, but learn a bit more about insects and some of the other parts of paleoecology in the ancient world that I'm really interested in.
I've never been in a practical lab like this before, and it's really cool to see everyone doing their research and stuff and what the research process is actually like. I've learned a lot about the equipment, like working more in depth with microscopes and with samples and stuff like that, and in other labs as well. Like we worked in the DNA lab, which is a much different environment than this lab here. It was really exciting looking through my samples and then finding something like an insect part or a little piece of plant material after searching through the dirt and the samples a lot. Getting them identified was also really exciting to me. It's been really cool to see what the research process is actually like. It's definitely increased my interest in research a lot. I enjoy it a lot, giving me more interest in maybe pursuing something like that in the future.
The specimens come from the Inuvialuit settlement region, so within their territory. Hopefully we'll have a meeting kind of towards the end of the internship to communicate that back to the community.
This is my third year having an ISTEM student, and this is our student, is Chance Park. He's investigating how the climate change, such as drought, affect the mycorrhizal fungal communities in the soil. We collected a lot of samples from a lot of places. After collection, we had to identify the fungal biodiversity inside the soils. Chance is contributing that aspect of the research project, looking at the DNA extraction, and then he's using this electrogel phoresis. He's quantifying the DNA.
We extracted the soil in the top layer and the bottom layer, mixed them together, went through processes to isolate the DNA. Once we get the PCR and DNA in here, we can take it and scan it, and it'll tell us how much the DNA is actually amplified and if we can send it off to get the different things be identified and such. I found it so interesting how isolating that DNA and adding different primers and how the enzymes and everything work and actually getting what you want. It's a very niche thing. That was so cool to me to learn that process. So many of our provincial parks and stuff have fire suppression and stuff like that, so mountain pine beetles are a huge issue. To bring these forest lodgepole pine stands, maybe to make them healthier, we can understand what these ectomycorrhizal funguses do. If we can identify what type of ectomycorrhizal funguses are in the soil, we can maybe inoculate or do certain things like that to enhance the growth.
Our goal is really how can we restore these forests back to normal. Studies so far suggested that actually mycorrhizal fungus and plus in the injection of these we call the extraction from the mycorrhizal fungi, it's really promoting the seedling health very well.
This program has really changed my mindset and made me really start thinking towards a master's and really heavily considering and wanting to. And also just the confidence of working in a lab and doing experiments, and this ecology lab is a base core of many Indigenous communities is having how ecologically everything ties in together, and how, for example, lodgepole pine and this fungus that lives in the soil associate, everything is connected in some ways.
This project in particular is studying organic agriculture and the effects of non-fertilized and non-herbicide treatments on the growth of various crops. Mason is one of our most eager and most driven interns in the lab. He is always willing to do whatever task and loves to learn about everything that we're doing.
I've learned a lot about the different ways they analyze soil and looking at the different nutrients through them. And so I've learned a lot about lab work when it comes to agriculture science. This agriculture research is very important for Indigenous communities because it's looking to find more sustainable ways to improve farming, which is very in line with keeping a balance with nature.
Some of farming and agriculture is lacking Indigenous representation, and so the pathway between Indigenous and, I guess, conventional agriculture or conventional farmers is really important, and he brings a lot of value to our project.
I've learned a lot through my short time at ISTEAM about the master's and PhD programs, and I'd say it's definitely influenced me. Considering now I would like to go into the master's program and hopefully do some research of my own someday.
Because the first experience was so great that I wanted to try to come back for a second term, and this is an opportunity to do something new, something different, and still have the same general sense of being in that lab setting on the U of A campus, which is a great opportunity to get in the first time. So I was extremely happy to be accepted back for a second term.
Eric's work in the lab involves making a video for our research project to be shared with farmers and organic agriculture associations. So his work is primarily with science communication.
My role has a lot more to do with the videography and communications of working going on in this lab. The focus of this video is a lot to do with Alessia's project in the sustainable agriculture and the research that goes on into it. Specifically to do with the reduction of greenhouse gas emissions and the increase of carbon sequestrations. It's extremely important that we look into these factors while still trying to figure out an organic agriculture alternative to the methods and techniques of farming today. So to get this video out and have the opportunity to create something like this and spread this kind of message is extremely important to me. It's a great opportunity to be hands-on and active in the environmental sciences fields, specifically ones that they might be interested in or in ones that they may not know as much. Having this opportunity to come out here into the field and learn all about this kind of stuff has been a wonderful opportunity. I think it's really great that the opportunity is afforded to not just U of A students, it's open to anyone that is interested in the environmental sciences field of Indigenous background. So if there is anyone out there that likes this kind of stuff and would be interested in the kind of stuff, it was definitely an opportunity you should look into.
I have it at a concentration of 10 millimolar.
Okay.
Just testing to see if it reacts at this concentration. So it's a pH of 7 again.
So you think this will be faster or slower?
I think that this one actually won't react.
Okay, yeah, that would be interesting too. We're really interested in how these chemicals that are in the environment can move through the environment and react away, and we're also really interested in if they don't react at all. The project this summer, working with my iSteam intern, Samantha, is studying this antioxidant chemical contaminant that's been found throughout Canadian waters. So in surface water, in drinking water, and we want to understand if it reacts with sunlight. So is that a way that the environment can naturally remove it? So what Samantha's working on here is with this photoreactor, studying how fast this chemical reacts away. It's called DPG, or diphenylguanidine. So she's doing some really interesting sort of fundamental chemistry, but applied to the environment so that we can have a better understanding of how this one specific contaminant exists and potentially threatens our water resources.
This study that I've been working on has been studying tire wear additives in aquatic environments. So it's a contaminant in our water systems, and it can help all communities and all people, especially sometimes the Indigenous communities depend on the land for their water and all their resources that is often not filtered. So focusing on water quality is very important for Indigenous communities.
Samantha already knew she was interested in this sort of research, but it's been a really great opportunity for her to connect with the Indigenous community on campus and learn more about Indigenous knowledge and thinking.
I always knew that I loved chemistry and I wanted to work in environmental sciences, so this has been an amazing opportunity for me to grow and expand in my field of work and find some new career opportunities. The iSteam program was really put an emphasis on grad school for Indigenous students. So I learned a lot about that and how the process works and what opportunities there are for grad students who are Indigenous.
For us, it's been a great way to combine our work with the Indigenous community on campus.
My most exciting moment was going to the CTAC PNC conference. It was the first conference that I was able to present at. I made a poster and I got to present my research to a lot of professionals in the field and peers. It gave me a lot of experiences and with professional development as well, and just told me all about the opportunities there are out there for Indigenous students.
She's well organized, and most importantly, she has this enthusiasm to do and complete her task and to learn new things.
I think this has been a great summer so far, and looking forward to future years, hopefully, of the iSteam program.
Okay, you ready? Okay. Put that just on top, as gently as possible. Try and just fold that over.
So it'll be good to just like identify them like that.
Yeah, we get back in the lab and identify any flea beetles on this one. If anything, I feel like that one right there, that would be a crucifer flea beetle. Kateri is looking at damage assessment caused by the flea beetles in canola using ImageJ to assess how much damage has been done by the flea beetles.
I learned a lot about insects and how they play a role in the ecosystem. I feel like I've learned more about lab dynamics and how to socialize more. Whatever I learn here, I could help my community with other potential future impacts that it may face.
Let's move on to the next.
10% this time. That would be best, right?
Yeah, 10 is about the maximum we have tried.
Mm-hmm. I think I'll do 3 grams this time because I might need a larger batch.
Sounds good. So this project is about studying the recycling of plastics. It is a fundamental study on how to use physical separation methods using solvents as a precursor to doing thermochemical conversion to recycle plastics.
Janaya is using a series of solvent to screen the desired reaction conditions, at what temperature, at what reaction conditions we can get a selective separation of these plastics.
This gives us both fundamental understanding of what's going on, but also a way to design a process that can work at scale.
I really enjoy this project when it comes to recycling. This is actually something I've always been quite passionate about, but I was definitely raised, to be aware of it. And, when I teach my nieces and nephews and things like that, I'm like, this is our earth. We only have one planet, we have to take care of it. I've learned to be a little bit innovative. Like, they'll ask me questions, they're like, what do you think you should do next? And I'm like, let's try this. And so they really give me a lot of free range for what we can do next. And so it's not just me being bossed around, which I really enjoy. We did lots of literature review so that I could actually understand what I was doing, which I really appreciate. I definitely think it's broadened my perspectives, made me realize that maybe I'm capable of things I didn't realize I could do. It's just made me a little more confident. I really enjoy this project, that I'm actually doing something worthwhile rather than working a normal job that is just a job. And so I feel like it has a really good purpose.
The program Linnea is working on focuses on the effective and secure use of subsurface resources for cleaner energy transitions. We were very fortunate that we managed to talk her all the way from Ontario to here to Alberta, at the University of Alberta.
Working in this lab with Dr. Gonzalo has been an amazing experience. We do a lot of research into land restoration post oil mining operations. I'm trying to find a way to use rubber as a replacement for silicone sleeves, which would give us the benefit of using extra sensors placed on the rock. I've just really liked the environment, and I feel like it's taught me so much about myself and also just understanding the scientific process here. It solidified my want to continue to pursue research. All my coworkers have really been encouraging me. They've always wanted me to ask questions and make sure I really understand why I'm doing what I'm doing and what the purpose is and the applications to the real world. That's kind of what I want to be doing for the rest of my life.
I'm working on putting together and programming a wireless system for monitoring conditions in an agricultural shipping container. This project is planned to be sent to northern communities struggling with inflated grocery prices, so it will help northern Indigenous peoples struggling with costs of shipped fruits and vegetables. I have learned about microcontrollers and sensors, wiring, 3D printing, and even lots about biology and agriculture. First time I got data sent from a temperature sensor to read on the computer screen through Wi-Fi was absolutely amazing. It felt like I'd just done some magic. This internship has helped me to realize there's so much more to environmental studies than just studying biology, and that biology doesn't have to be all blood and guts either. This program has been incredible. I've had so many opportunities to try things I wouldn't normally have wanted to try, like picking strawberries and harvesting and weighing plants. It's been incredible to branch out and experience different kinds of science that is more hands-on than anything I get to do at school. I'm really looking forward to future opportunities to work with sensors, bioinformatics, or maybe even botany.
The iSTEAM program has been incredible so far. I've learned a lot about programming. I've gotten to do a lot of coding in languages I would normally use. And I've been learning a lot about different things I can do with my career instead of just basing it on the classes I think. It's nice to get real experience.
We are out here at Coal Valley Mine in Alberta. In our project, we are in partnership with ClearFlow, who's looking at the management of effluents and the effects they have on water chemistry, fish toxicity, and invertebrates, in order to ensure that the water chemistry, the water management, and the invertebrate structures are healthy, and to test for any impacts. I would recommend the iSTEAM Pathways program if you want to gain insight into research, environmental occupations that help local or rural Indigenous communities.
And then we might
might get some metallic beetles as well, but I don't think we've been getting that many in recent catches. My research is looking at how mountain pine beetle disturbance. We're looking to see whether the mountain pine beetle has an impact on unintended organisms within that environment that they've wiped out the trees in. What Michaela is sorting here right now is from last week's catch, where we go to several sites around south of Grand Prairie. We have multiple traps set up to collect for different types of insects.
During this internship, I've been helping with different projects.
I feel like I've been a pretty big part of what Riley's been working on. When we go out into the field, everybody that's there kind of does every part, which is nice. Like I have experience with working with the pitfalls and panel traps and funnel traps. And then when we're in the lab, I'll sort all of those different types of traps. So I really get hands-on with all aspects of the research.
What's the smallest clam that you guys saw?
4 millimeters.
Oh wow. Let's see those baby clams, Brianna. Oh, that's very cool. They're all fluorescing. The projects that we work on are related to ocean change and how factors like climate change, invasive species, and habitat loss are impacting ocean species. What we've done is both experiments and fieldwork where we're looking at the types of species that green crab like to eat and what the impacts are on those populations.
During this internship, I've learned a lot of different skills on the computer and how to use Boris software. And I've also learned a lot of practical lab skills such as like dissecting and measuring and weighing different items. And I've also got to learn how a lot of different processes work and how different equipment works. And I think that I've also really learned how to be adaptable and how to ask questions, which I think is very important in both science and in research. This research is really important because invasive species, they decimate habitats and they really affect ecosystems. It's really important for First Nations communities because we rely on native species that are currently getting reduced in population because of invasive species.
It's been a real pleasure having Brianna in our team. She has been making all kinds of amazing observations of the projects that she's been working in. We wouldn't be anywhere near where we are as far with all of the information that we've gathered this summer if we didn't have Brianna working with us.
This one has higher nutrition compared to this one. Our goal is to unlock the potential of wild berries, and that's basically growing in abundance in the northern and indigenous communities. Hannah is very important for our project. She's collecting samples, learning how to characterize the nutritional quality using the high-performance liquid chromatography. She's also bringing the indigenous perspective to our work, actively participating and contributing to the overall objective of the project.
During my internship, one of the exciting moments I had was being welcomed into the NRC and given a tour, going into the laboratory, learning how to run the HPLC machine, watching how the machine runs the nutritional quality of berries. I'm so thankful to have this opportunity to learn and to grow as a person in the academic world. And I'm so thankful for the ICE team and my supervisors for everything that I've learned this summer.
This ICE Team Pathways program is fostering the inclusivity and diversity in scientific research. It's not only empowering indigenous students, but also integrating indigenous views in environmental research.
I feel like being able to feed this cell is going to hopefully reveal a lot more rich data this year. Okay, here we are. Jordan last year built some skills in 3D printing and designing of fuel cells. This year he used those skills to redesign and deploy a new type of cell, but also has been learning about more advanced data analytics techniques, like how do you analyze the data, as well as thinking through all of the different things that we need to do to have more information about water quality. This really has been a great skill building, like I said, looking into a little bit deeper about water quality parameters and what influences water quality in general, and using that knowledge then to inform how we could make newer technologies and newer sensors that add value. So we have a lot more clarity than we did last year just by the continued experience, and I think for me it's been really, really good this year. It's accelerated things greatly.
This year I designed second type of cell. This one is floating or semi-submersible cell, so it could go up to about a foot underwater. What this fuel cell will do is allow for real-time monitoring and collect data and be able to tell the biologists, researchers, or other people who are monitoring the cell information on what's going on with the water that they choose to monitor. iSteam and working with Adam for a couple years allowed me to get a further taste and develop my interests, and now I kind of want to try to pursue environmental robotics.
I absolutely love iSteam Pathways program. I think it provides perfect opportunities for students to learn about their field, about environmental science. It's about being a part of a community, and that really is important. That sense of shared goals and shared vision and working toward reconciliation ultimately. So iSteam is really, really an ideal program, and I'm happy to participate.
Working to try to find unmarked graves of children who never came home from residential school is an essential part of reconciliation in the lands we call Canada. This has become a very important issue over the past several years, with the news of many different projects underway to undertake this difficult work. The Institute of Prairie and Indigenous Archaeology has been asked to help many nations in searching lands using technology to try to find these sacred locations. And it is a really important part of healing and reconciliation, as well as justice and accountability for the children who are lost. One of the interns has been working directly on residential school projects, supporting the field work and the application of technology in community-centered and respectful ways, and having Indigenous students being able to come with us into community to learn about the process, what's involved, the various technologies that we use, and then how we make sense of the information that we're gathering is really important for capacity building, for getting those students engaged in the scientific process, but also learning to do so in very respectful and community-led ways.
I do believe that the research that I'm helping with can help Indigenous communities with healing, because the impact of residential schools still rings true and continues to intergenerationally affect Indigenous populations. I think that iSteam creates a lot of opportunities and pathways for Indigenous students in science, especially because there's a lot of barriers for Indigenous students in science. And I can say that for myself, it has helped me fight those barriers too.
So my intern, Sophia Jurio, has been working on supporting the Exploring Métis Identity through Archaeology project. And Sophia has been helping develop policy that will support Métis communities to protect and to steward our own heritage.
Heritage policy is underdeveloped throughout the majority of Canada. And when we lead with Indigenous leaders, we can ensure that our documents and material culture is protected. This internship provided an opportunity to expand on heritage policy for the Métis Nation.
I'm really grateful for the opportunity to have been part of this, and I really hope that this program continues, because it is really seeding that next generation of scientists and scholars with and for Indigenous communities.
Thank you to the iSteam Pathways program. Thank you for this experience.